nested hierarchies in constrained and unconstrained regions. Apparently, it's a direct refutation of the common design hypothesis. I had a lot of fun discussing this particular issue. So, Grayson, Jackson, anybody who wants to discuss this particular argument or challenge, you're saying that if, according to the separate ancestry model, the original created kinds are designed in a way that allows them to have internal diversity. As a result of internal diversity and basically DNA differences that are separating species and kinds, you are going to naturally find some kind of hierarchy. From that position, you would argue that over 6,000 years, more accurately 4,500 since the flood, since there was a bottleneck, those hierarchical patterns that were once there in unconstrained sequences, they should no longer be there because mutations accumulate in unconstrained sequences without any real selection. There's no purifying selection acting upon those regions. And so those regions should diverge more and more where there is no hierarchy seen if separate ancestry is true. Go ahead, speak to that. Is that more or less accurate?
Isn't there background selection, though, that selects against the ones that are harmful, but then also parts that are not really doing anything?
Well, great. Grayson,
I mean, you could have some selection effects just if they're close to like areas that, like, as a chunk, you can have selection effects in chunks of the genome. But largely speaking, in unconstrained sequences, there's not going to be purifying selection. So you would expect a pattern of mutations to be pretty stochastic, which is like kind of synonymous with mostly random.
Okay. My challenge is this, but also a question of curiosity. And in our discussion during the open mic, I don't feel like the evolution side adequately answered it, but I do really appreciate the technical back and forth. And so this is probably up for debate, right? So let's see. I don't believe that the evolutionary model does predict hierarchies after millions of years in unconstrained sequences, because these unconstrained sequences, there's no purifying selection, or there's very limited, as Grayson was saying. Remember, selection sees the really big mutations. Those are the ones that it sees. It can amplify the best beneficial mutations, but largely, and what we see in unconstrained sequences, largely what we see is an accumulation of neutral mutations. And so they're not subject to selection. And so they build up over time and they drift through genetic fixation, randomly for the most part. And some drift to fixation. Anyways, over millions of years, if we have all these mutations accumulating generation after generation without selection in these unconstrained regions, then today we shouldn't find any consistent hierarchical signature, because those mutations should have scrambled or muddled the unconstrained sequences to the point where there is no hierarchy. Can you speak to that, gentlemen?
Yeah, I can pick that up real quick. I think it's a basic question of numbers. So you and Matt like bringing up that CO1 gene study, right, with the DNA barcoding. I just want to point out, because I didn't get to address it in the last open mic, but I just want to address that is not talking about the sequence. That's talking about barcoding, which is like different things. But in that study, you point to basically the conclusions that they have that, that for most species, the CO1 gene kind of looks different to barcoding after like 200,000 years for like over 90% of species, right? So that CO1 gene is less than 700 base pairs. So basically you're kind of starting to get a different barcode after like 200,000 years for 700 base pairs. So then if you're talking about the total unconstrained regions, you're talking about billions of base pairs. So for 700 base pairs, it takes 200,000 years to get a different barcode. So for it to lose that pattern of nested hierarchy, I mean, just do the math. I mean, for 700 base pairs, it takes 200,000 years.
Right.
How much would it take for billions of base pairs?
So a 200 base pair region. Just a second. My wife's bringing me a bottle of water. It's right here.
Yeah,
Donny
it's
drinks a lot of water.
it's not perfect because it's DNA barcoding, and it's not
Right.
really it's not really looking at whether or not you can reflect nested hierarchical patterns off of that, but just as a proxy for to gain an understanding, you can look at that CO1 gene study, and
That's a
for
good proxy.
700 what?
I agree. That's a good proxy. That's
Yeah,
a good,
for 700 base pairs, you get a different barcode after about every 200,000 years.
yeah, that's
So
a good
then,
control.
yeah, so then if there's, you know, estimates are anywhere between 80 to 90% of the human genome is non-constrained. So that's out of what, 4 billion? So that's going to be only like, you know, 3.5 billion, let's just say, unconstrained base pairs. How long until you get until the nested hierarchical pattern disappears from that? I mean, it would be many billions of years.
Okay. Before I give you a strong rebuttal to that, I feel like I need to ask you a few clarifying questions just to make more sense of this argument. Unconstrained. What's your definition of unconstrained or unconserved?
Like if it's not under if it's not under the effects of purifying selection, like basically if that the actual sequence of the base pairs is not contributing to the function of that region. So it basically, if there's an equal chance of that base pair position being any of the 4 letters, like, then it would be unconstrained, like in a population.
How do you know if a region of the genome is under purifying selection?
I believe that there are genetic analyses that can be done to estimate if a region is constrained or unconstrained.
Are you looking at the amount of mutations that are accumulating in one region versus another?
Yeah, I think you compare, like, in a given population, certain areas, and like certain, you like look at certain base pair areas, and you can tell like how constrained or unconstrained they are based on the variance within a population. That's my understanding, but they could estimate them through other means. I'm just not sure.
I suspect some inherent circularity in defining unconstrained, because I hear evolutionists like you saying, well, unconstrained is a region of the genome that's not under purifying selection. But then how do you know if an area is under purifying selection? Well, because it's unconstrained. I feel like there's a circularity in the definition of it. Speak to that, Grace. I'm just trying to really clarify some of these points, but that's kind of what I'm gathering from the basic definition of it. It's unconstrained because it's not subject to purifying selection. How do you know it's not subject to purifying selection? Because it's unconstrained. It doesn't have as many mutations accumulating basically as compared to another region, it has more mutations accumulating than another region, which doesn't. Something for the heart or something. So speak to that briefly, Grayson.
I would need to look into specific studies of their methodology for how they're like operationalizing constraints. But I do know, like I just sent a study in the private chat that is like a genome-wide constraint map of the genome that they looked at for 76,000 human genomes. So I believe that they take the population, like a given population, like in this case 76,000, and they compare each and every position and look at the variance in the population. And the closer you are to having, basically random positioning of letters at certain locations, right? And this, remember, it's important to keep in mind that we're talking about individuals here, and genetic drift culls like 99% of individual mutations. So when you're talking about the patterns of nested hierarchy in non-constrained regions, you'd be talking about like more fixed, which is a population size effect. So when you're looking at individuals in a population, you can compare each individual region and see like the closer that they are to being, 25, 25, 25, 25% of each of the four letters. That means that it's closest to being non-constrained because it doesn't seem to have any impact which letter is in that position.
Okay. Now, how much of the non-coding regions? So basically, we have what 2% of the genome codes for proteins. Would you argue that most, if not all, of your protein coding regions of the genome are constrained?
Those are the most constrained typically in the genome are the protein coding regions, but the regulatory regions, any sequence where the sequence is important for its function is going to be more highly constrained.
Okay, so you would then argue, I'm guessing, a lot of your regulatory regions and so although the non-coding regions of the genome are more unconstrained, there are some areas within the non-coding regions of the genome that are constrained, like regulatory regions.
Yeah, that's an important point. So non-coding is not synonymous with non-constrained. They don't mean the same thing. So yes, you can have a non-coding region that is highly constrained.
Okay. Those clarifying questions help. So let me bring up a point here, and it's also somewhat of a counterpoint. So in order for forward evolution to work over millions of years, the neutral theory of evolution, don't you require enough mutations, neutral mutations over time to drift to fixation in order for enough diversity basically to accumulate in organisms for serious evolution to take place? You have all these neutral mutations accumulating. Eventually, they reach fixation and evolution progresses.
Well, if they're neutral mutations, then they wouldn't really be contributing that much to any fitness. But I mean, sure. I mean, genetic drift does happen, and genetic drift can fix neutral mutations for sure. It does.
So if you have, just throw a number, say you had 50, let's say the per generation neutral mutation rate was 50, right? As Fatuma says, since on average it'll take 4 NE, basically how long it takes for mutations to drift, neutral mutations to fix to be fixed in a population, generations for such mutations to reach fixation, about the same number of neutral mutations should be fixed every generation. In the evolutionary scenario, you have mutation drift equilibrium. Okay, but for the young Earth creation model, for the audience sake, because Grayson, I know you already know this, we don't have enough time to reach an equilibrium where we have a lot of substitutions taking place. I'd argue, yes, after the flood, you have 8 people, and for somewhere between 500 and 1,000 years, the human population doesn't spread out to all parts of the globe, so you can have some fixation there. But since that's spreading out, you've got no fixation. It's too slow. People are spread out. So, but in an evolutionary model and time scale, you have mutation drift equilibrium, and therefore, with the longer time scale, the number of neutral mutations per generation that accumulate, that same number equals your fixation rate. So say we had 50 neutral mutations per generation, the per generation rate, you'd also have 50 mutations becoming fixed per generation. So that would, as a result, over millions of years, muddle this hierarchical signature more and more, because that's a lot of mutations that are becoming fixed. When a mutation gets fixed, it's now stuck in place where every single individual in the population has it. If genetic entropy is true, and we're going to talk about this in great detail on Thursday, and those neutral mutations are more so nearly neutral, as in they're slightly deleterious, that's actually irreversible, unless you get a back mutation, but it's pretty rare, but irreversible damage in that population, because that nearly neutral mutation is now fixed where everybody has it. There's no going back. It's not going to be removed by selection. It's not going to drift out of the population. And so, yeah, I would argue this problem, if there is a problem, is a bigger problem for the evolutionary model because of how many mutations would become fixed per generation in an evolutionary time scale. Speak to that a little bit, Grayson.
Yeah, so I think that you brought this up with Dan on the last stream, and he pointed out that this is sort of an idealized math right here, and it's for haploids, and it's assuming like random mating, or there's all kinds of assumptions baked into that statement from the introductory textbook there. So I can take it as like an approximation, but again, it's not going to be very exact because those assumptions that go into that math are obviously not true. So,
but is this theory true or not? Like, do you agree with this basic population?
I just addressed that.
I said it sounds like you're saying, well, it's true, but it's not really.
It's an approximation, but it's like not going to be, it's not going to be accurate in real life because we're not haploid. Recombination happens, non-random mating happens. There's a lot of things baked into that math in the assumptions that it rests on that are just not in reality true. But it can be a fine approximation.
Well, I would contest that it's
a larger problem, even if it's all true and it's exacting. I think that it's still a problem of not understanding the sheer numbers involved. I mean, how many neutral mutations become fixed each generation out of 4 billion base pairs? It's not going to be that significant when you're looking at the total numbers.
Well, so if the rate of fixation of neutral mutations equals the neutral mutation rate, if it's 50, if it's 100, if it's 10, that's how many are becoming fixed each generation. But over the course of 1 million years or millions of years with the evolutionary time scale, that's a lot of episodes of irreversible or nearly irreparable damage where basically these
neutral.
Right. And that's the question. Are they completely neutral or are they nearly neutral where they're only slightly deleterious? I'd argue if they're nearly neutral and slightly deleterious, slightly harmful, then that makes evolution impossible. Yeah, you're going to have to argue that they're completely neutral. There's no deleterious effects.
So what's that? What you're saying is called negative epistasis, which means that all of these slightly negative effects add up to be a large negative effect. And that's not observed. Like, we haven't observed negative epistasis happening. I mean, it's just something that we don't observe. These seem to be, for all of our observations, neutral mutations that are not adding up to have some. When you have enough neutral mutations, suddenly you have a big negative effect. Like, that doesn't seem to be happening in real life.
But I feel like there's a few points here that I want to address, though. Before I ask you this, I want to point out that Dr. Dan, and again, I appreciated his time during that discussion. I thought it was excellent. When I showed him the quote, he did say, okay, it applies to diploid organisms, but the math is complicated. And so this is the theoretical basis of the molecular clock, as Fatuma says. But when you start accounting for various things, then it becomes more multi-dimensional. Okay. But then when I asked him if the math has been done specifically on humans, he said he hasn't done it, and I haven't seen anybody that's done it yet. So I don't know how impressive this evolutionary response is that is basically, well, yes, this is true, but when you actually do the math, it's more complicated. Well, where's the math? Where's this non-existent math that makes it more complicated?
Well, you're taking this from like it says right there on the screen, an introductory level textbook.
Yeah, it's basic population genetics, right?
Yeah, why don't you go to a graduate level textbook and see what it says there?
Well, because this is like, for example, when evolutionists say, oh, well, I mean, you're looking at the per generation mutation rate. You're supposed to look at the fixation rate. It's like, dude, basic population genetics says that your per generation neutral mutation rate equals the fixation rate. So if it is 50 per generation, then 50 are becoming fixed per generation in an evolutionary time scale. I would argue that that time scale isn't there, but if you want that time scale, you do have this problem, especially if these neutral mutations are actually slightly deleterious or nearly neutral.
How is
which
it a problem, though?
Because if you have 50 slightly deleterious mutations being fixed every generation, then that's irreversible or irreparable damage. I understand there's back mutation, so let's say nearly irreversible damage in the genome. That damage is now stuck in place. There's no way to get rid of it unless that population goes extinct. But that's extinction now.
Well, individuals can go extinct. Well, will die, not extinct is the wrong word. But
well, I wanted to ask Grayson this: how do you interpret Kimura when he says under the present model, effectively neutral, but in fact, so to me it sounds like he's clarifying what effectively neutral means. Very slightly deleterious mutants accumulate continuously in every species. So when Kimura uses effectively neutral, isn't he defining that as being slightly deleterious?
I don't think so.
Well, then what is... I mean, he says under the present model, effectively neutral. What does that mean? In fact, very slightly deleterious mutants accumulate continuously in every species, and here's the rate of loss of fitness per generation may amount to 10 to the 7 per generation. And so, yes, if 50 effectively neutral or nearly neutral mutations are accumulating every generation, and that amount are also being fixed in the evolutionary timescale, that is irreversible, irreparable genetic damage population-wide.
Here's something I don't get about this. They have to be neutral or nearly neutral to accumulate unseen, but as soon as they become harmful, they're going to be selected out.
But then they're not neutral anymore. But then they're not neutral anymore, so it doesn't apply.
What I'm saying, as soon as it causes a bad effect, and it's going to cause a bad effect in all of the population at once, it would cause a negative effect in an individual with a hypothetical.
And that's a great question. That's the challenge. That's the problem, is selection so limited because selection involves the death or removal of individuals. And so if population wide, you have 7 billion people that have accumulated all these nearly neutral mutations, and every generation basically is worse off than the previous generation. Okay, like you said, enough mutations can accumulate where the damage is large enough for selection to see. Well, what are you going to do, Jackson? Are you going to just remove the entire human population? That's extinction. Well, how much of the population is going to be removed?
Not all humans have this. Humans don't have all the same mutations. So, that totally changes the equation, right?
Every generation, you have roughly 100 new mutations per person per generation.
Right, but not all the same.
You have 7 billion people. That's 700 billion new mutations this generation alone. The human population has experienced every possible mutation in one generation.
Right. I was reading a paper, and I don't have it. I don't remember the exact title. I have to pull it up, but I have it somewhere. It said deleterious mutations accumulate faster in bigger populations, but their mean effect is diminished, and they're more effectively removed. Right,
but that's the thing, is by the time a population, okay, another way to call this is carrying capacity or even mutation count mechanism. By the time a population reaches a point where the genetic load is so extensive that Mother Nature selection can now see it, that population has descended into such genetic sickness that the only way selection can save the day is basically extinction. But extinction's not good.
Here's what I don't understand about that. What I'm saying is not all humans are going to have the same mutations. The same mutation rate, yes, but not the same mutations. So as soon as something negative builds up in one, maybe that individual would die, and a more fit, so to speak, genome person would continue to live, and it would just kind of reach an equilibrium, right?
The problem with that, Jackson, is it's a misunderstanding of how genetic entropy works. The point is, yes, there is fitness variation in the human population. Okay, some individuals have more deleterious mutations than others. But it's that every single generation as a whole is more mutant and more worse off than the previous generation. Like me today, you today, we're more mutant than we were yesterday. We're more mutant than we were 10 years ago. And so, yes, there is fitness variation, but nonetheless, population wide, every single human being is multiply mutant, and every single human being is more mutant than the previous generation. And so if you were to invoke, for sake of argument, unrealistic selection, you removed 50% of the population, but it was only 50% of the worst mutants, those that actually have the most deleterious mutations. So you got rid of, say, 4 billion. Well, you're still left with 4 billion people that are more mutant than the previous generation. And so even though you've removed the worst from the equation, those that you're left with
All right, let's say
are
that
still
happened.
worse than the previous generation. Does that make sense?
Let's just entertain this and say that happened. 4 billion people with horribly harmful mutations died, and 4 billion people with pretty much neutral mutations lived. And then the population would get back to that level, 8 billion again. And then the process would repeat again. And that kind of reaches an equilibrium.
That's a great point. But the problem is, a great point to engage, that is. So you're left with the 4 billion people that are still more mutant than the previous generation, right? Well, now that population continues to grow where you get another 8 billion people, however many years that takes place. Well, those individuals now, say 100 years later, whatever it is, they're still way more mutant than the generation was 50 years before that, or 20 years before that, or 10 years before that. So yes, selection can certainly slow down the process, but it's never actually going to solve the process, because again, every generation will be more mutant than the previous generation. And eventually a population will descend into such genetic sickness where you're basically just going to get a mutational meltdown. It's tough because eventually a population reaches a point where it kind of, it becomes more of a, not that it's less of a problem, but that population can stay in that condition for, an extended period of time, but they're staying in a condition that is essentially mutant.
Donnie,
does that make sense?
My screen?
Yeah.
So I would just want to point out that what you're saying is not true if there's such a thing as neutral mutations, effectively neutral mutations. And I found the rest of that quote, as you can see here. Sorry. As you can see here, okay, my computer's kind of, okay. This is the quote in its context. So first of all, I want to point out that he's not talking about all effectively neutral mutations here. Like when you read it in the context, he's talking about different issues that could potentially arise. And again, this is in 1979. Further work has been done. But in this original paper, he's talking about there could be effectively neutral mutations that are in fact very slightly deleterious, that can slip under that threshold of being visible to selection, right? So he's not talking about all effectively neutral as you're trying to make it sound, like when he's saying that you're trying to make it sound like he's saying all effectively neutral mutations are in fact very slightly deleterious. He's not saying that. It's not.
Well, no, I'm not saying. I'm saying when he uses the term effectively neutral, that's what he means. But
that's not what that is not the case. That's what I'm saying. That is not the case.
Well, where does he say that?
Under that big umbrella of effectively neutral, you can have very slightly deleterious mutants. That's not to say that all effectively neutral mutations are very slightly deleterious. He's not saying that. Like, he's saying that these very slightly deleterious mutants can fit under that umbrella of effectively neutral and kind of smuggle in, which is what you're saying, that this can build up. That's what he's talking about here.
Well, and that's why
you have to read until the end where he says that whether this small rate of deterioration of fitness constitutes a threat to the survival and welfare of the species is a moot point, but it can easily be taken care of by adaptive gene substitutions. Like, he answers the problem right there. You just only were quoting the first part.
Well, I appreciate you pointing that out, but I'll go back to the quote that I had. I have that part in there, and notice here. Oh no, wait, right here. whether such a small rate of deterioration in fitness constitutes a threat to the survival is a moot point. This will easily be taken care of by adaptive gene substitution. So he's saying it can be, but he never actually did any work. He never provided any math. He never ran any simulations to.
Do you know that for a fact?
Demonstrate. Yes. He never examined this argument or this assertion basically comprehensively, but also.
Do you know that for a fact, though? Like, are you just saying that he didn't do that in this paper, or you're saying throughout his whole career? Because he's a very established geneticist.
Well, where
You're
did
saying
he?
that he never did that. He never constructed any mathematical models showing that to be the case.
I'm not going to go on record and say that he never did, but I predict that he hasn't, because I haven't seen anything presented where he ran a sophisticated analysis or he provided any evidence backing up this claim that adaptive gene
substitution. Have you specifically looked for the evidence?
Yes, yes, I have. Yes, I have. And so, and it's not found. He makes an assertion, and maybe he's hoping other population geneticists actually look into that, but no sophisticated analysis of this assertion has been done. I can be corrected if somebody presents me with.
I definitely would like to hear Zach Hancock see if he agrees with you on that.
Okay, but I wanted to point out a lot of this as well depends on how much of the genome is functional, because I have a feeling Kimura is saying yes, those effectively neutral mutations that are very slightly deleterious are those that are occurring in, say, a non-coding region of the genome that's maybe functional in regulation or something. And so they're going to have to maintain, I believe it was Kondrashov as well, who argues the genome is no more than 10% functional in order that the majority of the genome can actually absorb these neutral mutations, making them absolutely neutral and not slightly deleterious. But if more of the genome is functional, say 50% of the genome is functional, well, that's now 50 nearly neutral mutations accumulating every generation and not just neutral, as in with no effect on fitness. I mean, Iyer, Walker, and Kitely here says this. It seems unlikely that any mutation is truly neutral in the sense that it has no effect on fitness. All mutations must have some effect, even if that effect is vanishingly small. I mean, yeah, I'd agree with that. All mutations must have some effect on fitness. Now, whether he's referring to functional regions of the genome, let's say in the non-coding regions, speak to that a little bit. But I would agree that, yeah, no mutation can be considered absolutely neutral in the sense that it's got no effect at all. No, I would argue that each mutation, most of them having only a small effect.
Yeah, but what do you mean by small effect here? Because I mean, we've talked about this exact thing before, and I mean, if you read the rest of their paragraph, they talk about what they mean by effectively neutral, and it's totally fair. But if you really wanted to consider like some sort of, if you wanted to look, I mean, they even say vanishingly small, not just small, vanishingly small effect. So if you really wanted to consider like on a minute scale the impact of the fitness of every single mutation, well, obviously to produce each letter of the gene of the DNA, like the A's, the C's, the T's, the G's, is going to have a slightly different biochemical cost in terms of the amount of ATP and resources it takes to create like a purine or pyrimidine. Like these are different chemical compositions. So obviously they're going to have different energies involved in creating them. So even if you just have a mutation from like, an A to a T, well, suddenly you've changed the total amount of energy cost by a very, very, very small amount, but a change. So yeah, obviously you can calculate that it has an effect on fitness, but it's so small that it's negligible for all intents and purposes.
I would argue that a single spelling mistake in a book the size of an encyclopedia, that is vanishingly small. But enough of those typographical errors or spelling mistakes accumulate over time, then that book is no longer readable.
Right, but if 90% of that encyclopedia is not sequence, like it doesn't need to be in a specific sequence, like an unconstrained region.
Right. See? And so it comes down again to junk DNA. If the majority of the genome is junk, let's say only 10% is truly functional, then yes, you're going to say that the majority of these mutations, say the mutation rate's 100, only 10 of those are going to be nearly neutral or effectively neutral, as in slightly deleterious, because the rest of them are landing in positions in the genome that's just neutral junk. And so it has no effect on fitness. So a lot of this rises or falls on junk DNA. Now, I will say Mendel's accountant, which I understand you're not a big fan of,
Nope.
but they've at least run simulations where they assumed that the majority of the genome was junk. And the results were that even if the mutation rate was somewhere between 1 and 5 per generation that were nearly neutral, then genetic degeneration was inevitable. So it appears that even if the genome's 10% functional, genetic degeneration is still inevitable. But
Yeah,
again, that's going to rise or fall on a debate dealing with Mendel's accountant.
So
but
two things. One, the consensus among geneticists is that junk DNA is very real and exists and can be demonstrated to exist. That's the consensus among the experts in the field of genetics. Two, speaking specifically about Mendel's accountant as a model, I want to just ask you to be skeptical, and I want to ask you to look at the assumptions that go in to the math of Mendel's accountant and ask yourself if those are reasonable assumptions to make.
Okay, I want to point out a couple things. Since we just argued extensively about junk DNA, we're gonna leave that for now. Okay, obviously you believe most of the genome is junk. I would argue that most of it is functional, and we just engaged that for a while the other day. But going back to the unconstrained regions and the hierarchies that they fall out into, okay? My first point, I would contest the claim that this is predicted by evolution, right? You've responded, you've given some points to consider. But the points that you've made equally apply to the creation model, because firstly, our time scale is only 4,500 years. Those unconstrained regions comprise a lot of DNA base pairs. Yep. They're massive. Again, only 2% of the genome is protein coding, 98% is non-coding. I understand not all of those non-coding regions you'd consider unconstrained, but most is what I remember you saying. And so
it's like 80 to 90% of the genome is non-constrained.
Okay. So in 4,500 years, we're not going to have substitutions. The neutral mutation rate is not necessary. We don't have the long evolutionary time scale for there to be this mutation drift equilibrium where you basically have your neutral mutation rate equaling the fixation rate over time. So we don't have those instances really where you've got irreversible damage, where you have all these mutations becoming stuck in place or fixed. Yes, after the flood, I do believe we have rapid fixation during that time period. But once people spread out to all parts of the globe, well, the genetic fixation is going to be very rare. So my point is, we only have 4,500 years for mutations to accumulate in your unconstrained regions. 4,500 years is nothing. You're not going to get a significant alteration in those hierarchies that were there at creation. You might get some change, but you're not going to get a whole lot of change.
Have you seen that study on the substitution rate measured at Tristan da Cunha, an island in the South Atlantic that was settled by the British only a couple hundred years ago? Like maybe it's 300 years that, and they've measured the substitution rate, and it matches the phylogenetic substitution rate, like measurable. And we know when the first people showed up on that island, only like max like 300 years ago, and we've measured the substitution rate in that population. It's a small population,
right? But I'm not. Are you trying to say that because that's 200 years? In evolutionary time scales, you'd have your neutral mutation rate equaling the fixation rate.
Donny, are you saying there's not enough time for fixation to happen?
No. In In the evolutionary model, you have the long times, you have millions of years basically for mutation drift equilibrium. And so therefore your rate, your per generation rate where neutral mutations accumulate, they will equal your fixation rate. So if you have 50 per generation, you have 50 becoming fixed basically. Okay. But, and remember those unconstrained regions, there's basically no selection. Grayson and Dr. Dan argue limited selection, but the whole argument against the creationist model depends on those unconstrained regions not being subject to purifying selection. But when I point out this challenge that there shouldn't be a hierarchical signature if the evolutionary model is true, now suddenly different forms of selection are invoked. It kind of sounds like the
that's just
evolutionists
not
thought their
true,
cake
though.
and eat
Like,
it too.
no,
Yeah,
like
well,
the
Dr.
objection.
Dan was looking to the linkage blocks. Remember, he said if a deleterious mutation accumulates in one of these large linkage blocks that have accumulated neutral mutations over time, now selection removes that organism from the equation.
Yeah,
That's
he's just
selection.
trying to be careful, like with his statements. Like the entire, like for the majority of neutral mutations,
Right.
like it's probably not going to be the case. I mean, for the majority of mutations, they're going to be neutral and they're not going to be under selection in non-constrained regions. That's for the vast majority. There are always exceptions. This is biology. There are literally always exceptions to everything you learn
Right.
in biology.
And I agree, the exception
So,
doesn't overturn
so
the rule.
Dan's just trying to be careful of making absolute statements in biology when there are always exceptions. So he's a scientist. He's trying to be accurate in his statements, but that doesn't mean that you can say with any supporting, there's no support to say that in evolution we would not expect for the non-constrained regions to show a nested hierarchy. That's absolutely
Well, I'm not okay.
a prediction.
I understand what you're saying, but I don't think my point's being heard. The point is, if over millions of years you have unselectable mutations accumulating every single generation, unselectable meaning they're not subject to purifying selection. Okay, if that is not going to alter the hierarchical signature, then 4,500 years since the flood of mutation accumulation in unconstrained sequences is also not going to alter the hierarchical signature. Those hierarchies that were there at the flood are still going to be there 4,500 years later, even if several mutations have accumulated in unconstrained regions of the genome. That's my point.
But the hierarchies of the mutations wouldn't have any reason to be there.
There's not. It's 4,500 years of mutations in a model that doesn't have mutation drift equilibrium, where you basically have hypothetically 50 nearly neutral mutations accumulating per generation, and therefore 50 being fixed per generation. We don't have that issue. And so, yeah, 4,500 years of mutations accumulating in regions of the genome that comprise how many millions of DNA letters? It would take a long time to alter.
DNA letters.
Right. Yeah, exactly. So it would take a long time to alter those regions to the point where there's no longer a creationist hierarchy. But I mean, the whole point of incomplete lineage sorting, the whole point of examining various different gene trees, right? Depending on what you look at gives you a totally different tree. Aren't you arguing that, yeah, over time, mutations have accumulated in a way where, depending on the gene you look at, especially in unconstrained regions, might actually put, a human and a gorilla closer to a pig than to an orangutan? I'm just throwing an example out there.
Like, you have to look at the total. You can't just look at one or two genes
exactly right
and make your comparison. You have to look at a broader amount than just, looking at one or two genes.
Right. And you're doing that because mutations have accumulated and accumulated at different rates in different people groups, or different organismal groups have had different histories, different patterns in terms of mutation distribution. And so you have to look at a larger repertoire of genes rather than just cherry picking a single gene that may have mutated differently across the biological world. And so now you have an inconsistent hierarchy. You're saying that's not enough. You got to look at the bigger picture, right? You got to look at a big set of genes.
Yeah. When you look at the big set, then you start to see the phylogenetic relationships.
And that's my whole point. My whole point is the creation model doesn't have this struggle or this challenge because we only have 4,500 years of mutations accumulating in these unconstrained regions that number in the billions of DNA base pairs. There's not going to be a massive alteration in hierarchies is my point. I don't see it as a challenge, especially if it's not a challenge to the evolutionist after millions of years of unselectables accumulating and building up over time. Does that make sense?